Good day, class. We are at Unit 4 of our biochemistry subject, and this is all about amino acids and proteins. Proteins came from the word or the Greek word proteos, which means first rank of importance, and this macromolecule mediates bioprocesses or every bioprocess in the cell, such as cell metabolism, cell growth, and neurotransmission. There are 20 major amino acids that is needed for the formation of proteins. So now we proceed to the learning objectives of this unit. So first is at the end of this unit, you shall be able to describe the structure of amino acids and determine their classification. Second is to explain how amino acids are linked together. The third one is to analyze the different levels of protein structure and describe how proteins are denatured. And the last one is to distinguish the different functions of proteins. Now we go to the first part of this unit, which is amino acids. Amino acids share common structural features. They are colorless, non-volatile crystalline solids and can be denatured at above 200 degrees Celsius. The general structure of an amino acid contains four functional groups. Namely, the first one is the carboxyl group, which is the COOH, followed by the amino group, which is the NH2, and a hydrogen atom, and lastly, the R group or the side chain, which is a unique or which gives the unique characteristics of the amino acid in terms of solubility, size, shape, and ionic characteristics. And lastly, it also confers the identity of the amino acid. The R group is unique for every amino acid. One of the unique characteristics of an amino acid is having an acid-base nature. So also, they exhibit both negatively charged and positively charged regions. So the COO minus here is the negatively charged region, and the NH3 plus here is the positive region. Also, amino acids are predominantly dipolar or Zwitter ions. Zwitter ions are compounds which have no overall electrical charge or with a negative or with a zero net charge, where this is designated as its isoelectric point. Amino acid reactions are therefore very pH dependent. At a change of pH, amino acids tend to accept or donate protons. The Zwitter ionic form explains the physical properties of amino acids, which what I mentioned a while ago, that amino acids are crystalline solid. They have very high melting point and they have high solubility in water. Another properties of amino acids is, except for glycine, is having a chiral carbon center which allows stereoisomerism. This allows the functional groups of the amino acid to form a tetrahedral arrangement. So this is the tetrahedral arrangement. In addition to its being chiral, the amino acid structure contains four different groups of atoms bonded to the alpha carbon. So that is what we discussed a while ago. Also, the structure is optically active. Just like your left and right hand, this pair of amino acids does not have a complete or a perfect overlap. These are called enantiomers. Why is this important? So this allows the amino acid to rotate to the plane of polarization or to have an optical activity. So we have here an example, the L-alanine and the D-alanine. So the D and L configurations are absolute configurations around the chiral carbon. In here, the the nature of the side chain dictates the magnitude of the direction and rotation of the amino acid. Dextrorotatory or the D behavior refers to the clockwise rotation of the incident light. On the other hand, levorotatory or the L behavior refers to the counterclockwise rotation. So this is another example of stereochemistry, which shows the D and L configuration. We have the L-glyceraldehyde, and the other one is the glyceraldehyde with the T behavior. It is also important to note that amino acid residues in naturally occurring protein molecules are exclusively L isomers, and D amino acids are found only in a few peptides, example of which are bacterial cell wall and some antibiotics. We now go to the classification of amino acids based on nutritional requirement. So the first one is the essential amino acids. So these are amino acids that must be supplied in the diet since they cannot be synthesized by the human body. Examples of which are tryptophan, isoleucine, phenylalanine, valine, leucine, methionine, threonine, lysine, histidine, and arginine, while non-essential amino acids can be synthesized by the body. Examples of non-essential amino acids are glycine, alanine, proline, serine, asparagine, glutamine, tyrosine, and cysteine. As mentioned earlier, the nature of the R group or the side chain dictates the properties or the characteristics of the amino acid. Now we go to the classification of amino acids based on the structure or the functional groups in the side chain. So we have here 7 classifications. So the first one is the alkyl or aliphatic, which is the nonpolar. And then we have the sulfur-containing alcohols and amides, which are the polar uncharged, the aromatics, the acidic classification, which is the negatively charged, and the basic classification, which is the positively charged. So the first one is the alkyl or aliphatic, which is nonpolar. So they stabilize proteins through hydrophobic interactions. So examples of alkyl or aliphatic are the glycine, alanine, and proline. So you can see here they are nonpolar. And then proline limits structural flexibility because of its rigid ring. Other examples of alkyl or aliphatic are leucine, isoleucine, and valine. So as you can see here, the R groups are all nonpolar. So the sulfur containing is here, examples of which are cysteine and methionine. So they are very hydrophobic. Cysteine is responsible for disulfide bridges in proteins. So as I said a while ago, sulfur containing is an example of a polar uncharged. Then the next one are the alcohols. So the alcohols, example of which are threonine and serine, they are also examples of polar uncharged. So they stabilize protein and interact with the aqueous environment through hydrogen bonding. Aromatics, on the other hand, examples of which are phenylalanine, tyrosine, and tryptophan, stabilize protein through hydrophobic interaction. They can participate in hydrogen bonding because of the OH in here. The negatively charged groups are called the acidic, okay? So they have a COOH side chains which are ionized at pH 7. So as you can see here, they are negatively charged. Examples of acidic classification are the aspartic acid or aspartate and the glutamic acid or the glutamate. The next classification is the basic group, which have which has the NH3 plus or the positively charged region. Okay, so examples of which are lysine, arginine, and histidine. So they have the NH2 or NH3 plus side chains, which are protonated at pH of 7. Again, this group has positive charge. The next group is or are the amides. So amide counterparts of acidic amino acids. They can stabilize protein by hydrogen bonding. So these are also polar uncharged groups. So example of amides are asparagine and glutamine.